Welcome to the summary video for the Introduction to Viruses lesson. Viruses are one of my favorite parts of microbiology. To me, they're so fascinating because for something that's not alive, it has such a tremendous influence on our body, on plants, on fungi, on even bacteria. Different viruses have evolved to parasitize pretty much every type of cell that has ever been found. Every type of organism can have a little virus parasite infect it. So you will begin this lesson by learning about the fact that viruses aren't alive. They are not made of cells. We'll talk about virus structure, or you'll learn about virus structure, and what little there is to speak about virus structure, at least as compared to prokaryotes and eukaryotes. Here is just a schematic of just a very, very small handful of the viruses that infect humans. These parasites that take over our cells and use all of our machinery and our materials to copy themselves. Yes, if there were no other living things, viruses could not survive. They are completely dependent upon hijacking a living cell and using the cells to make more of them. So, virus structure. Well, different viruses have unique things about them, but generally, there are very few parts that are found in all viruses. All viruses have some form of nucleic acid that carries the instructions to make them, just them, okay? So viral nucleic acid is quite small compared to prokaryote, especially small compared to eukaryote DNA, because there's just doesn't take many instructions. There's not much information required to make a new virus. They are so small and so simple compared to living cells. So viral nucleic acid is how we classify our viruses. We talk about RNA viruses or DNA viruses. And then within that, we talk about the number of strands of the nucleic acid. So you can have a single-stranded piece of DNA or a single-stranded piece of RNA, a double-stranded piece of DNA, or a double-stranded piece of RNA. So if you're an RNA virus, you'll have to do certain things within the cells and use certain machinery to multiply. If you're a DNA virus, you'll have to do other things and use other piece of machinery. So we speak about them in generalities based on the nucleic acid because that really determines the basic steps that the virus is going to have to undertake in order to replicate itself, to duplicate itself within our cells. Okay, so all viruses carry a piece of nucleic acid. That's the instructions to make them. All viruses have a shell, a protective shell around that nucleic acid called the capsid shell, okay? And the function of the capsid is literally just to protect the nucleic acid from the environment around it. And then all viruses have glycoprotein spikes that stick out. And different viruses have different spikes that are unique to that virus. And these spikes you will see are the keys that allow them to get inside of different cells. They take advantage in animal cells like us. They take advantage of a natural process called endocytosis, where the cell is used to receiving environmental signals. And the spikes on the outside of the virus mimic these normal environmental signals. So the cell thinks it's getting its regular molecule bound onto its receptor. And the regular thing to do would be to take that molecule that was bound onto the receptor and bring it inside of the cell. Well, what the virus does is it mimics that regular molecule using its spikes. So when the spike binds onto the cell's receptor, the cell thinks it's a normal thing and it brings the virus inside of the cell. So the cell hijacks this normal endocytosis process to get inside, and that's one of the major ways that viruses enter cells. But you'll learn more about that in the videos and in your readings. So there are three basic structures that are found in all of the cells that infect humans, all animal cells: the nucleic acid, the capsid, and the glycoprotein spikes. One option that you find commonly is a membrane that surrounds the capsid. So if you have a virus that has the membrane, we call that an enveloped virus. And if the virus does not have the membrane, then we call it naked. So if it's just the capsid with the spikes and nucleic acid, no envelope, then it's naked. If it has the envelope on top of its capsid, then the membrane, it's called the envelope or enveloped virus. That membrane actually comes from the cell that it was made in. So you inhale in a virus, it attacks your throat cell, gives you a nice sore throat. And as the virus makes new babies inside of your throat cell, it steals some of your own membrane to coat itself with as it leaves your cell. So it cloaks itself in you to distract your immune system from attacking it. So that's the basic structure of an animal virus. You will learn about the multiplication cycle of an animal virus. It attaches onto a cell, it gets inside of the cell, it opens up its capsid and exposes its information, its nucleic acid, and then it copies, it goes through the normal information processes where the nucleic acid is translated into protein, and those little virus proteins all get assembled into new baby viruses. And then the last step would be that the new baby viruses leave the cell and go and infect new cells. The point of a virus, the virus is simply a parasite whose job is to multiply. It's just to reproduce itself and spread from one cell to the next, to the next, to the next. So that's what the virus multiplication cycle is all about. And you will learn the steps, the basic steps of it in this lesson. The other major kind of big picture thing with this lesson would be comparing prokaryotic viruses and eukaryotic viruses, particularly animal viruses. Plant viruses and fungal viruses have slightly different approaches to them, and protist viruses because of unique features of those cells. So we're focusing on humans in this class, so we are going to compare bacterial viruses versus animal viruses, human viruses. So our basic animal virus we just talked about, we'll do a naked one. Here's our little spikes. A bacterial virus basically looks just like that on the top, the capsid with nucleic acid, but instead of having spikes, it's got these big, long, leg-looking things sticking off. So here is our animal viruses, the ones that infect humans. Here is a bacterial virus called a bacteriophage, or phage for short. Right? And the reason that it's got these strange appendages sticking off instead of just using spikes is because of the anatomical difference between eukaryotes and prokaryotes. So animal cells, remember, just have a membrane on the outside, and we've got these little receptors the virus attaches to, and ta-da, inside of the cell. Well, bacteria have their membrane, but they also have their cell wall and potentially another membrane on top of that. And bacteria don't do endocytosis. They don't have those receptors. They're not used to communicating with other cells in their environment, so they don't have that same type of process that the animal viruses use to get in. So they can't do that little trick. So what they do is they punch a hole in the outside of the cell. So part of this bacteriophage apparatus, these legs, there is a cool little kind of spring-loaded drill almost in this neck part of the bacteriophage that the bacteriophage will land like an alien on the outside of the bacteria, and it'll punch a hole inside of the bacteria and release its DNA. And then that DNA will be picked up by the bacteria's normal cellular machinery, and the viral proteins will be produced. So it's got a slightly different structure because it's got a slightly different way to enter into the cell. And all of that is because the structures on the outside of the bacteria versus eukaryotic cells are different. And the mechanisms, the processes that are happening within the cells are a little bit different. So they had to evolve to have a different way to get in. The final major comparison between bacteriophage and animal viruses comes into the concept of latent, for animals, viruses, and lysogeny versus lytic for bacteriophage. Lots of L words in this particular lecture. So a latent virus is one that gets inside of the cell, so it enters inside of the cell. Got your little virus in there. And then it just sits there and waits. It's waiting for an environmental factor that tells it that the immune system is weak. When the immune system is weak, then that virus will continue its multiplication cycle, making new baby viruses, and then the new baby viruses will erupt outside of the cell and releasing lots and lots and lots of new babies into the environment. So what would be the advantage of waiting for the immune system to get weak? Well, if the immune system is weak, then the little babies that are created are less likely to get destroyed and more likely to infect new cells and be successful. So that's what latent viruses do. Think of like herpes. You get super stressed out by a job interview and suddenly a cold sore appears. Well, that was because the herpes family of viruses have the ability to be latent. Once they get inside of you, they can crawl inside of your cells and sit there and wait. And so you don't know that you have an infection because they're not actively multiplying and causing damage. Instead, they're waiting for your immune system to get weak before they emerge. And then they go back into hiding again. So you get it and you have it for life, even though you're not constantly showing symptoms. Bacteriophage do kind of a similar thing, or at least they have a similar option. So lytic phase is when the bacteriophage inserts its DNA inside of the cell, and then the bacteriophage immediately reproduce. They make new baby bacteriophages. The cell rips open. The babies are released. So that's like the immediate replication cycle. That's what lytic is. Lysogenic is when the bacteriophage enter inside of this cell and wait, much like latent for animal viruses. But instead of the nucleic acid just sitting inside of the cell waiting, what the nucleic acid does is it actually inserts itself into the cell's piece of DNA, of nucleic acid. So at this point, the virus is literally part of the cell's genetic makeup. So it's not just sitting in there silently waiting, it actually becomes part of the DNA. So if this cell multiplies, the new baby cells that will be made will have the virus DNA inside of it. So it's an advantage for the virus because it's sitting there protected without, and can actually be multiplied and spread to new cells without having to do anything. However, if that original cell gets sick or starts feeling, strain from the environment, the food source starts dying or the pH gets wrong, and that original cell starts showing signs of stress, then that bacteriophage, that virus, will pop itself back out of the DNA and will start multiplying and will release. So it does not die, it does not sink with the ship if the ship starts going down. So lysogeny is a pausing step, much like latent is, but the bacteriophage actually insert themselves into the cell's DNA and become part of the cell's DNA during that lysogenic period. And that only happens with bacteriophage. There are very, very few examples of animal viruses that actually insert themselves in or have that phase where they're actually part of the cell's DNA. It's not impossible. It happens. HIV is a famous example of that, where there's kind of an insertion stage. It's just not nearly as common as you see in bacteriophage. And the technical details are a little bit different, so you don't use the term lysogenic for those rare examples of that insertion in animal cells. So I hope this lecture is interesting. It's really setting the stage for us to start digging into a bunch of different viral diseases that we'll see for the rest of the semester. As always, if you have any questions, had any stumbling points, please post in the Muddiest Points discussion board, or send me an email or talk to me in lab, and we will hopefully answer your questions. Good luck.